Thank you for the introduction. And thank you for inviting me to present our work at this trainee recap among the fantastic talks that have happened so far today. I'm excited to share our abstract with you looking at white matter hemodynamics across the adult lifespan, and I have no financial disclosures to declare. So why are we interested in white matter hemodynamics? We already know that structural integrity decreases with increasing age. For example, fractional anisotropy, or FA, globally decreases with age, as shown in this study on the left panel. We also know that white matter hyperintensities arise with increasing age, as seen on the right side of this panel, and are a sign of neurodegeneration. Taking a look at the frequency distribution of white matter hyperintensities compared to the distribution of FA values, we can also see on the bottom part of this figure that the frequency distribution of these hyperintensities is far more skewed across the aging population in comparison to the distribution of FA values. This indicates that there isn't necessarily a clear picture of how white matter structure changes during aging and what drives the differences between typical and atypical aging. Additionally, studies have shown a clear relationship between white matter vasculature and white matter pathology. For example, this study used histopathology to identify reduced white matter vascular density compared to the overlying cortex. They found that white matter capillary width correlates strongly with white matter lesion score in various neurodegenerative disorders. This could be a compensatory change to retain white matter perfusion and integrity during hypoperfusive states related to the age-related dementias. It's therefore theorized that changes in physiology may precede some of the structural abnormalities that occur with neurodegeneration and aging, but the relationship between structural integrity and cerebral hemodynamics remains unclear. There are numerous existing barriers to white matter perfusion imaging using arterial spin labeling, or ASL. Firstly, the maximum SNR as a function of resolution for different arterial transit times, or ATTs, is much lower than that of grey matter, such that longer transit times result in lower SNR for a given resolution. We know that white matter presents longer transit times than grey matter, given the depth required for blood to reach white matter. Second, longer post-labeling delays are also necessary to account for the longer ATTs present in older populations, as shown in this table. Bold values, highlighted in red here, indicate statistically significant differences between young and older populations for an ASL acquisition with vessel suppression. There's also a lot more variability present in transit time values across the aging process, and this ultimately means that multi-delay ASL is a more beneficial approach, but is not widely used in large cohort studies. As a result of these barriers, studies in white matter in large, typically aging populations have been limited. One study used a single delay pCASL sequence to study global white matter hemodynamics and found that cerebral blood flow, or CBF, generally increased with age. They also found elevated CBF in women compared to men, with the strength of the relationship with age for CBF varying based on the lobe investigated. Different regions may therefore present different hemodynamic characteristics. And our group's previous study, in a group of 254 participants from the Lifespan Human Connectome Project in Aging, or HCPA, which employed state-of-the-art multi-delay pCASL, also found distinct results. So the trend of cerebral blood flow globally in white matter was nonlinear with age, and arterial transit times was associated with increases with age. So this highlights the importance of disentangling the effect of ATT in CBF estimation, which is possible with multi-delay acquisitions. In general, women still demonstrated elevated CBF, as seen in the previous study, but by using an approach for quantifying CBF that corrected for hematocrit values, we saw a smaller effect of sex. So this array of findings led us to the following research questions. Firstly, how do white matter structural integrity and hemodynamics correlate in typical aging? How does sex influence white matter structure and physiology? And are these effects region or vascular territory specific? We used the wider HCPA cohort to investigate these findings, which consisted of a typically aging cohort, excluding for any suspected major illness. There was also no exclusion based on medication use to ensure good representation of the aging process. Generally, there was a similar age distribution across each biological sex. The data collected in HCPA includes 2D multi-delay pCASL with 5 post-labeling delays between 200 milliseconds and 2200 milliseconds, and a labeling duration of 1500 milliseconds. RTR was around 3500 milliseconds and T of 19 milliseconds with a high spatial resolution for ASL. And here you can see an example of some of the raw images collected. The diffusion data included 4 runs of opposite polarity encoding directions, 1.5 millimeter isotropic resolution, following B values, and 3200 millisecond TR and 89.2 millisecond TE. And again, you can see some of the examples of this data here. So for the diffusion data, we use top-up for distortion correction and eddy for motion and eddy current correction. We then imported this data into DTI-FIT to calculate fractional anisotropy. A lot of this was done using the FSL or fossil toolbox. For the perfusion pipeline, we again corrected for distortions using top-up and included in-house multiband and motion correction. We then modeled ATT using a cross-correlation approach that we've previously described and used the ATT value we extracted in a 2-compartmental CBF model that accounts for age and sex effects of hematocrit. Finally, we projected our ATT and CBF maps onto the white matter fractional anisotropy skeleton to identify the effects of CBF and ATT more specifically at a tract-based level. So first, we looked at the average measures of CBF across the cohort, where lighter red indicates higher CBF and darker red indicates lower CBF. The average CBF appeared elevated in the anterior portion of the white matter compared to the posterior portion of the white matter. But for ATT, we found longer ATT values in the posterior portion of the white matter and body of the corpus callosum. So we think this could be related to potential arterial stiffening in the posterior circulation, as these flow territories may be more susceptible to neurodegeneration. Here we see the association between CBF and age, where stronger negative effects are shown in lighter blue compared to darker blue. In general, CBF decreases are associated with increasing age in white matter of the body of the corpus callosum. And for the association with ATT and age, stronger positive associations with age are seen in the lighter colors. So as you can see, ATT increases are associated with increasing age, and this effect is widespread across white matter. Overlaying the two relationships with age, there are clear differences in the spatial distribution of CBF and ATT associations with age within the white matter areas. It could be that by measuring the time it takes for blood to reach a particular region, we are more sensitive to the effects of vascular tortuosity, arterial stiffening, and other similar measures of hemodynamic compromise. The relationship between fractional anisotropy with age demonstrated a negative association in the majority of tracts, but a positive association was observed in the longitudinal fasciculus area. On the one hand, this could be a neuroprotective effect of healthy aging, which has been seen in prior studies. However, we can't ignore the potential contribution of crossing fibers in these regions, so more work is definitely necessary to investigate this effect. For sex differences, it was once again clear that CBF is elevated in women compared to men and vice versa for ATT, with stronger differences shown in lighter colors. The effect of sex was more widespread for ATT than CBF. However, we did not see widespread differences in fractional anisotropy between sex, which could indicate that these physiological differences we see are independent of structural changes, and that structural changes may be more strongly associated with age effects. So looking at the correlation between CBF and FA for each tract, a clear relationship was evident between the two measures. Here's an example of one tract in particular. When we computed these correlations voxel-wise, we saw this relationship was also widespread across the brain. However, this correlation was much stronger both in specific regions and across the brain for ATT and FA. So again, this could be an indicator that ATT may better predict changes in structural integrity. So to summarize, the effects of age vary according to vasculature surrounding the white matter tracts. Longer ATT may be a better indicator of hemodynamic compromise. Sex differences that we observe here agree with the existing literature. And there's a clear correlation between white matter structural integrity and cerebral perfusion. But future work should investigate the influence of lifestyle factors, cognitive ability, and medical history to aid further characterization of the aging process. So thank you for having me today, and thank you to all the funding agencies and my colleagues that helped make this work possible. I'm happy to take a few questions now. Otherwise, please feel free to reach me by any other media of choice. Thanks.